discussion,
Well, welcome everyone. Yeah, let me take a moment to get started here, and really thrilled for such a great turnout. My name's Kevin McGraw, professor in the School of Life Sciences, Danny's committee chair, and yeah, these are bittersweet times for advisors like me, where we've shared, in a lot of good times, a lot of good science. But these good things must come to an end. And with this defense seminar today, I hope you'll see that it has been a really exciting journey that Danny shared with us in the past 4 or so years. Yeah, so from friends, family, former lab mates, good to see community members. So much of Danny's life, it looks like, is present in Zoom or in the room. And that's always, for me, a neat thing to see, and a real testament to the support that he's received, throughout his dissertation. So he joined us in fall 2019, right? And well, it hadn't been that long. Too many of you will remember, well, frankly, a pandemic hit within the next semester. And in turn, Danny and colleagues, contemporaries, faced with this inordinately challenging situation for crafting a dissertation in the face of this unanticipated global event. Some people seem to think that we need to give, dissertations completed through the pandemic an asterisk, and let me be very clear that this is not one of those circumstances. And I hope you'll see through the work that he's done, the results he's uncovered, the journeys he's had, that he's really truly made outstanding lemonade out of the lemons he was dealt, and really cleverly putting together a series of studies that doesn't fit in some ways the kind of traditional mold of, in my lab at least, an organismal biology, single species sort of investigation. He's got a real clever compilation of studies, including some organismal in focus, but also nice comparisons between 2 clades of birds, and even with genomic and phylogenetic comparisons. So in sum, I think Danny's been a real effective, very creative scientist. I think he's really helped support the lab and boost, everything from morale to the quality of science. And so in those ways, I think we'll be sad to see him go, but we know he has an excellent offer under his belt. And so with that in mind, hopefully there's exciting, and in our colorful world, we think of things as bright, a bright future for Danny as well. So the way this song and dance goes is that Danny's here to present his dissertation research to you, roughly in the order of about 45 minute seminar. Public part of the presentation will continue with sort of a general question and answer period from the group. And again, I think we have a Zoom moderator to help field questions from online, as well as we can do it in person. So if you haven't grilled them adequately over those 10-15 minutes afterwards, we'll ask the public to leave the room, and then the dissertation committee will be left to ask questions from Danny and his work, finish up hopefully in a few hours thereafter. So committee have any questions at this point before we launch and turn things over to Danny? Well, I think we're ready to rock and roll. So without further ado, take it away, Danny Jackson.
I'm putting meeting controls.
Well, my name is Danny Jackson, as most of you know. I'm excited to share with you some of the insights I've gotten from my dissertation research into speciation and evolution in birds. I'm very grateful today for my committee, Dr. Kevin McGraw, who you all just heard from, and Gru Amdam, and Dr. Karen Swayze, and Dr. Scott Taylor. Thank you all so much for all of your guidance and support through this whole process. And I'm just very, very grateful for all of that support and excited to share these findings with you. So here I am in my full fieldwork gear for one of the studies that I did, the very scraggly beard, on desert cardinals in Tucson. And one of the core concepts that I want to talk about today is speciation. So just to kind of give you all a framework to understand what we're talking about, the process of speciation is the mechanism underlying all of the diversity of life on Earth, where a single lineage diverges into multiple lineages over time, which is how we have diversity of different organisms on this planet. And of course, these lineages can go on and diversify further and split into further lineages, creating many, many diverse and interesting organisms. But one specific aspect of it, so this is a very clean, clear diagram of it, but it's a much murkier process in reality. And we're going to focus in on some of that murkiness today. That's where a lot of my research is focused on is some of the unclear boundaries within speciation and trying to advance our understanding of this important process. And I address this in two systems. One is a highly mobile species or highly mobile clade of organisms, seabirds, looking at in the kind of absence of absolute barriers, where do we see gene flow? And then secondly, looking at how human activities, so how we are going out into the world and changing these evolutionary processes. And I use three different approaches to ask that question. But first, chapter one, looking at a clade of tropical seabirds, the main question that I asked is just what absolute barriers to gene flow exist. So these birds are wide ranging, they're able to fly anywhere. We think of speciation traditionally, or most simply, as a process in which a physical barrier creates an absolute stop to gene flow, small mice on either side of a big mountain range. But in species where that isn't quite the case, do we still observe gene flow both across land barriers and between species? And these are the 6 species that we'll be looking at. Blue-footed boobies, Peruvian boobies, masked boobies, Nazca boobies, brown boobies, and red-footed boobies. Hopefully, most of you are all at least familiar with one of them, blue-footed boobies. And yeah, they're all really charismatic and beautiful birds with interesting facial coloration and foot coloration and plumages. And 3 of them are wide-ranging throughout the tropics. Red-footed boobies and masked boobies and brown boobies are all found pantropically, so anywhere throughout the tropics. And you can see just right off the bat that there are 2 major land barriers to this distribution, the continent of Africa and the Americas. And we also then, since these are seabirds, have to consider physical processes outside of just terrestrial barriers. And one of them in particular is the Peru current or the Humboldt current along the coast of South America in the Pacific Ocean. And along this region, we see 3 endemic species, blue-footed boobies, which range from the Gulf of California, the Galapagos, and the northern coast of South America. Peruvian boobies, which are found along the kind of the southern extent of the blue-footed boobies range, and then further south to central Chile. And Nazca boobies, which are really just endemic to the Galapagos for the majority of their breeding, but they do sometimes breed on other islands in the Pacific. I'm going to try to hide this again. Okay. So this diagram of speciation that I walked you through earlier is the best predicted relationships between these species, with blue-footed and Peruvian boobies sister to each other, Nazca and masked boobies sister to each other. The 4 of those forming a clade with brown boobies as the sister to that, and red-footed boobies as the most anciently diverged lineage. There's been phylogenetic analyses of these with mitochondrial data and with multi-locus data. We look at this first with our whole genome sequence data, but prior to 1998, in that first mitochondrial study, Nazca boobies weren't included in it because we didn't consider them a distinct species. Instead, they were considered a population of masked boobies. So they weren't distinct species, but you can tell that there's some morphological and coloration differences in them. And then in 1998, they started, there was evidence that these should be considered different. They're morphologically different, behaviorally different, and the study found that they are reproductively assorting. They're mating only with other members of the same species. So this is one of the kind of murky areas that we'll be investigating with my data, and then further genetic evidence for the split came with mitochondrial data in 2002. Then also we see hybridization between a lot of these birds, blue-footed and brown boobies here, the parental species on either side, and then hybrid individuals that were confirmed with genetic data in the middle. Hybridization between Peruvian boobies and blue-footed boobies along the overlap in their range with, again, genetic evidence, and then multiple instances of interspecific courting or hybridization without additional genetic evidence. So these are putative, but yeah, we've seen blue-footed boobies and Nazca boobies courting, and they're putative hybrids from our colleague Dave Anderson, who noted that the Nazca boobies in the Galapagos sometimes have unusually blue feet. Masked and brown boobies with, again, putative hybrids between those two, just from field observations. So those are the different instances that we'll be investigating, but I keep referring to intergression, and I showed you that hybridization also, there's evidence of those, but these are distinct processes with different evolutionary consequences. And it really, the difference between these two terms comes down to whether or not this hybrid individual, which parental species in the solid circles up top, reproduce together and create this hybrid individual. The central question with intergression is, does this hybrid individual go on to reproduce, and with whom? If it doesn't reproduce, think a mule, like a donkey and a horse. It's most often sterile and doesn't go on to reproduce. That's just hybridization, but in the event that this individual goes on and reproduces with one of the parental species, then we see intergression, where some amount of genetic variation that's unique from this distinct species lineage finds its way into the species population of the other lineage. So intergression, gene flow from one species into another. And to address this question, I used whole genome sequence data. First, I confirmed phylogenetic patterns. I tested for gene flow between species and then gene flow within species across land barriers. And this is a map of all the individuals that we sequenced. You can see that we sampled widely throughout the ranges of each of these 6 species, and then ran different tests of all the instances that we showed you, or that I just showed you, of instances where individuals either hybridize or attempt to, or might be, or yeah. So first, how did we do this? I'm gonna walk through that with Nazca and masked boobies. We use an ABBA-BABA statistic. It's a test for intergression. Imagine that the most common recent ancestor of all of these 4 taxon has a genotype of A at a particular locus. If there are no mutations, all of their offspring will also have A. But if there's a mutation in this lineage, they would have what we're going to call the derived genotype of B. And in the presence of intergression, this mutation will find its way into this population. We can use a statistic called the ABBA-BABA statistic to compare rates of ABBA-sites, which occur in the presence of intergression, to BABA-sites, which are the null kind of hypothesis. And I'm not going to walk through all of the details of it, but just kind of a grounded sense of the way that we're able to test this. In the absence of intergression, we would see ABBA and BABA-sites at the same approximate rate, but in the presence of intergression, we see more ABBA-sites than BABA-sites. Very fun word to say that I practiced a lot. So first, we reconstructed the phylogenetic tree of these species and found that the species do, with our whole genome data, follow the same species relationships with blue-footed and Peruvian sister to each other, masked and Nazca sister to each other, et cetera, which allowed us to run the tests that we'd intended to. So looking at Nazca and masked boobies, you see that Nazca boobies are sympatric, or they overlap in range with masked boobies in the Pacific Ocean, which allowed us to then ask, because masked boobies are forming clades based on ocean basin, whether or not Nazca boobies share more genetic variation with masked boobies in the Indo-Pacific than they do with masked boobies on the other side of this land barrier in the Atlantic and Caribbean. So using that ABBA-BABA statistic, we were able to test for that and found significant evidence for intergression between Nazca and masked boobies. We then asked it for each of these following pairs, blue-footed and brown boobies, and found no evidence of intergression there. We found evidence between blue-footed and Peruvian boobies and between blue-footed and Nazca boobies, but not between masked and brown boobies. So the general trend that we're seeing there is that this 4-taxon clade, given the opportunity to hybridize, they still do. So there are some other barriers present there that keep them from converging, but beyond this 2.1 million year mark, there might be enough genetic variants that are incompatible that have accumulated between lineages. So this brown booby lineage seems to not be capable of producing introgression with any of these other taxons. Okay, so now that we've established that gene flow occurs across species boundaries, our next question was, does it also occur across land barriers between populations of the same species? And we asked that for masked boobies across the continent of Africa and the continents of the Americas. And we did find evidence in both situations. And then further evidence from red-footed boobies found patterns associated with gene flow across all tested land barriers. And then finally, for brown boobies across the continents of the Americas, we also found evidence for gene flow across those. So in this clade of 6 widespread seabirds, we see gene flow between species and across species. So gene flow is really, really widespread in the system. I think that makes it an interesting, this is interesting context for future studies to investigate some of the other barriers that might be contributing to the speciation process aside from land barriers in the system. So those are the main findings, and that allows us to move on to the kind of second stage of this dissertation, where we're looking at the effects of human activities on Arizonan cardinals. Looking specifically at urbanization, this is Tucson on the bottom and a desert cardinal up above, looking suspicious. This research fits into the broad context of the ways that human activities are reshaping our world and affecting ecosystems globally. This is a map of the regions considered the last of the wild. So the green areas are areas that have been have not been directly affected by human activities. You can see that here in the southwestern United States, there aren't really any areas coded as last of the wild, and that 77% of land's been directly impacted by human activities. Additionally, we've also lost 29% of migrating North American birds since 1970. This is total birds, not species. So we're seeing broad effects across this entire taxonomic group of birds, and I think that that sets the stage for the importance of this research and of other research in this space, highlighting the importance of identifying stressors that are affecting the wildlife in our area so that we can hopefully better integrate into our local ecosystems. Here in the Southwest, we see an 18% decline of birds. So urbanization is this massive change in the landscape. It redistributes resources in ways that aren't really seen anywhere else. We see novel patterns of water availability, novel vegetative communities, novel predators, and novel, especially in Arizona, heat stressors, artificial light as a stressor. And generally, we see declines in species richness across urban areas, especially if you've seen any of Jeff's research in the back. And then we also see the trend that generalists persist better than specialists across species. But these are really broad trends that have been characterized, and my research is trying to fill this gap at more of a fine scale question. What fine scale differences between species explain their ability to persist in an urban ecosystem? So in a situation of 2 closely related species with a very similar ecological niche, does urbanization differentially affect their spatial distributions is the first question that I asked. And these are the 2 species, Pyrrhexia, desert cardinals. You can see these are just observations of the birds, so not like a proper spatial model, but just points on a map where people have seen them. They range throughout the Sonoran Desert in Arizona, and they overlap really similarly with the range of northern cardinals. This is a distinct subspecies of northern cardinal that we have out here in the Southwest, and where they are distributed throughout the entire range is really, really similar from Arizona into Mexico with Pyrrhexia. So very similar. And then also at the very fine scale, they're occupying the ecosystem really similarly. This is the only other spatial ecology work done on these 2 species here in Arizona. The master's thesis from 1960. You can see in this kind of ballpoint pen figure that the two species, the main takeaway, cardinals on the left and Pyrrhexia on the right, are establishing overlapping territories with each other and nesting in very, very similar areas. So these two species, in the absence of human intervention, are occupying a very similar ecological niche. Which allowed us to then ask, does human activity, does urbanization disrupt this close relationship? And then what mechanisms can we identify that might be driving any differences we observe? This is the study site of Tucson, Arizona from above. You can see just on a first pass that we've really reshaped this valley with our urban landscape. On the northeast corner, you'll see the Santa Catalina Mountains. I'm going to refer to that a couple of times to kind of ground you in some of the spatial figures that I use. But this area has been inhabited for a very, very long time with irrigation and agriculture in the valley for thousands of years, but the extent of urbanization is really, really novel and very rapid and very recent. From 1950 to 1960, the population of Tucson expanded by 368% and has been growing very rapidly ever since. So this is a very rapid, very large stressor to this and redistribution of resources across this ecosystem that these birds have had to adapt to. To test for this difference in spatial distribution, I first use a set of statistics that ask yes, no, do they differ in their distribution or not significantly? And then the same question for their ecological niche. I modeled their distributions using a software called Maxent that I'll get into in a moment, and then looked into some of the mechanisms that might be underlying differences using a comparison of variables in the software, and then also testing for differences in means of the variables in our model. Maxent is the software that outputs this beautiful kind of heat map of the predicted probability of an organism across a landscape. And it uses environmental variables and species observations, which are, for my model, carefully chosen. I used elevation, principal components of climate data, and then distance to land cover variables. This is the one that we'll kind of delve a little bit more into in the results, but National Land Cover Database has this file where every pixel is coded as one of these categories. And I modified them to demonstrate or to create a raster file showing distance to each of these. So northern cardinals might rely on open water, but they're not going to be found on open water. They'll be found near open water. So we looked at all of these variables and then additionally modeled urbanization using a percent imperviousness file, which if you imagine downtown Phoenix, there's a lot of concrete, a lot of asphalt, a lot of apartment buildings. These are all impervious landscapes, impervious surfaces, and that's a way of approximating urbanization in a quantified way. And then I additionally used a canopy cover model and median household income to represent the distribution of resources that are socioeconomically determined. And then, of course, I needed observations of the birds, and I was very lucky to work with data that has been collected long-term in two different long-term data sets. Tucson Bird Count, which is run through the Tucson Audubon Society, has been collecting data for decades, and this is a structured bird count across the city. And then the CAP-LTER here in Phoenix also collects long-term data on with their bird survey. And then additionally, so those are both located within the urban areas, but I wanted to model it broadly across the state of Arizona as well. So I used eBird data, which is a community science platform where anybody can upload observations of birds to it. And then I can look at, use those observations in my model. So the main findings, these are a set of statistics that I ran to just test whether or not they have different distributions. The first three asked, do they differ in their distributions, and found significantly across all pest statistics and across Tucson versus Arizona that they do significantly differ in their distributions, but they don't differ in their environmental niches in any of the test statistics except for one that looked across the state of Arizona. So the general pattern here is that they're very similar in their sort of environmental variables that they're relying on, but there's some difference, some slight difference that's allowing one species to occupy more space than the other. So you see across the state of Arizona, northern cardinals in purple have a much broader range, especially across the northern part of Arizona, and Pyrrhexia are located around like Tucson and further south for the most part of their range. These are the models that Maxent output. And then looking specifically at Tucson, These are also models. You can see the kind of white triangle on the top right is the Santa Catalina Mountains in our model, where the two species are elevationally excluded. They do differ in their distributions across Tucson, but a better way of visualizing it, I subtracted the Pyrrolexia model in green from the model of northern cardinals, and then developed a statistical method to identify which pixels are significantly different. And this is that model. So you're only looking at the sites where the predicted occupancy of an area for northern cardinals is greater than that of Pyrrolexia, and they're all purple. I don't know if you can see it as well in the projector, but here we're showing that northern cardinals have a greater probability of occupying the urban area than Pyrrolexia do, and not so much of a difference along the kind of rural areas outside of this urban center of Tucson. Okay, and then we wanted to get a little bit into the mechanisms underlying this difference. So we asked how northern cardinals, which are found in both Phoenix and Tucson, what variables differ in their association with this species in the two cities and which ones don't. Interestingly, they don't differ between the two species in their association with imperviousness, which is our urbanization metric. But northern cardinals versus Pyrrolexia in Tucson do significantly differ in their imperviousness. So the same species in different cities is occupying a similar extent of urbanization, while different species, even though they're closely related and occupy very similar niche, do differ in their association with urbanization in the same city. And then further, northern cardinals between the two cities, they differed significantly in every single other variable that we looked at. So elevation, all those climate variables, et cetera, except for two others besides our surface imperviousness, which were distance from shrub scrub, also a very hard phrase to say, and percent canopy cover. So we're showing that these two environmental variables are very important for this species in their ability to occupy an urban area. And then comparing northern cardinals and Pyrrolexia in Tucson, we found that northern cardinals are found at closer to open water and at higher levels of both impervious surface and canopy cover. And Pyrrolexia, however, demonstrate greater affinity for shrub scrub than northern cardinals. So this difference, where northern cardinals are closer to canopy or higher canopy, higher percent canopy cover, but Pyrrolexia are more closer towards shrub scrub land, is kind of the main potentially driving difference between the two species, where when you think of how we're crafting the landscape across an urban area here in Arizona, we remove a lot of the native shrub scrub, a lot of the mesquites and the creosotes, and then we bring in a lot of artificial canopies, a lot of introduced trees that we pump a lot of water into. So that slight difference could potentially be driving this difference in these two species. And then also, yeah, I couldn't have done this without all these long-term data sets. So very grateful for that and for the role of open data in this. But that then sets the groundwork for the next chapter, where we're asking how urbanization is affecting the birds in their morphology. So looking at the plumages and then also the sizes of different traits in the two species to see if they're responding similarly across an urban area or not. I collected birds in the field and then also from museums, and I just collected measurements from the birds in the fields, not the actual birds. But Tucson Audubon helped me connect with a bunch of homeowners across Tucson to do this work in their backyard, and then used a lot of museum birds. Ended up with a sample size of 48 northern cardinals and 56 Pyrrolexia that range from 137 years ago until today, and are sampled across various extensive urbanizations. So from the urban area, from the outskirts around the urban area, and then from a lot of rural sites as well. Visually, you can see the green, the darker pins on the left are all of the rural sites, and then the kind of yellow-green are the urban outskirt sites, and then the purple pins are just urban bird sampling sites. So of course, since we're measuring traits across time, I needed across 137 years, urbanization has changed a lot in that time, and I needed a way to attribute a metric of urbanization across time. Luckily, Somebody else modeled urbanization during this project for me at a decadal scale. So these are raster files of whether an area is urban or not urban, with the black spots being urban. And I was able to use these to attribute an urban score to all of the samples dating back, 137 years to today. And then I measured several color traits. I looked at the color of the bill, the color of the crest, and the face mask of all of these individuals, and then also several size traits, looking at the crest, the wing, the tail, the tarsus length, the bill length and width at the nares, and the head length. You can see on the left, these are the birds that I caught and measured and released in the field, and then the museum birds here on the right. And our main findings were that while urbanization is not always associated with changes in the same traits, the effect is consistent and in a consistent direction for all similar traits. So for example, looking at male northern cardinals and male pyrrhexia, male northern cardinals, their breast is a red carotenoid-based plumage, and so is the crest of male pyrrhexia. So similar traits, and the effect is the same on both of them. So we're not comparing breast to breast or crest to crest, but different regions of the body. But in both of them, the urban birds have evidence of lower carotenoids in the plumage compared to rural birds. So similar effects on different but similar traits. And however, when looking at the bills of male northern cardinals, which are also carotenoid based, but have a different mechanism underlying that process, the bills are consistent with more carotenoids in the bill in the urban area, which is in contrast to what we're seeing in the plumage. So interesting effects on signaling traits. The bills and the feathers are all used in mate choice and interspecific signaling communication. Then we also see effects on maneuverability in their feathers, where male northern cardinals across time in urban areas are getting longer and longer tails, and male pyrrhexia and female pyrrhexia, actually just male pyrrhexia across time, regardless of urban area, are also getting longer tails, and female pyrrhexia across time are getting, their wings are shorter and shorter. All of these traits, both wing length, shorter wings and longer tails, are associated with increased maneuverability. So we see, again, similar effects, even if the traits are different. And then looking at the bills, we see that urbanization and time are both associated with larger bills. In male northern cardinals, bill length is longer over time, regardless of urban area, but bill width is only affected in urban areas where it's getting wider over time, both in male northern cardinals and male pyrrhexia, but male pyrrhexia, the bills are not longer over time. So this has potentially complex explanations, where the bill is used in a variety of ways. It's used in song, it's used in foraging, and the explanation that I think is potentially most likely is that it's used for heat dissipation. So we see a lot of urban heat extremes in Tucson, and a wider bill allows the birds to dissipate more heat. So that might be what we're seeing here. In conclusion, species respond to urbanization in similar ways, but not always the same traits. And it has complex effects on traits involved in signaling, heat tolerance, foraging, and maneuverability. And that sets the groundwork for the final chapter, which asks, do we see urban-related parallel evolution and introgression at the genome level? And I want to first kind of introduce you to why we thought this would be an interesting question to ask. In the absence of human activity, we might see a fitness landscape like we see here on this model of ants, where the two parental species have a high fitness compared to the low fitness of the hybrid offspring. But human activities reshape fitness landscapes. They make different traits more beneficial or more detrimental, because it's a completely novel context for the species to have to exist in. And we might expect to see a change in the fitness of the hybrid offspring in ways that might allow for more hybridization and potentially more introgression. And some of the evidence from my previous chapters are in line with this, where we see that one of the two species is less affected by urbanization than pyrrhexia. And we also see in the morphologies that northern cardinal phenotypes, both species are more selected for the phenotype of northern cardinals in an urban environment, that they have larger bills and longer tails, which northern cardinals have compared to Pyrrhexia. So the conditions are right for introgression between these two species in this direction, northern cardinal to Pyrrhexia. And we wanted to test that or see if the two species might be undergoing parallel evolution, where they're both responding similarly. This instance where gene flow might occur from one species into the other in order to respond to an ecological disturbance is called adaptive introgression. And we do see evidence of hybridization, not necessarily just in the urban area, but we know these birds can hybridize, although there's no genetic evidence of it, but there's just some goofy birds that have kind of intermediate morphologies and eBird data, and through the Arizona field ornithologists, they've published photos, and then also the Arizona Sonoran Desert Museum has a hybrid in captivity. So to ask this question, to investigate this, we sequenced the genomes of 6 urban birds and 6 rural birds of each species, and then ran a set of statistics to identify regions that are consistent with positive selection on that region. And then we identified genes that have had functional changes on them to further subset this list. So just looking at a phylogenetic analysis of these sequences, we see that the northern cardinals are clustering together and Pyrrhexia are clustering together, and there's no urban birds and rural birds are not forming distinct clades. So there's no evidence of population structure between them. But when we look at some of the statistics that we ran, these are FST plots of urban versus rural birds, northern cardinals above and Pyrrhexia below. We start to see some peaks. Each of these points in these graphs represents a region where the statistic was run on it across the genome. And then the higher points, the higher of the statistic, the more evidence for positive selection that we would see in the urban area. And we identified several interesting regions in both. And most interestingly, we saw that one region overlapped between the two species. It's this region with the gene CH037. And then, several other interesting genes just from the FST. And then again, we ran several other statistics, which prompted us to ask, how are these regions where they might be showing similar effects? Do we observe evidence consistent with introgression between the two species? And this is just a principal component analysis on the genes themselves, where we see in this plot that CH037, that interesting peak where they overlap. Urban birds from both species are clustering at the center of the plot, and rural birds of both species are separating out, suggesting that the urban birds have very similar genetic variation. And then we looked further into it and did a phylogenetic analysis on some of the genes and looked at, again, showing that rural Pyrrhexia separate out in these regions, but urban Pyrrhexia cluster with all northern cardinals, urban and rural northern cardinals, which is consistent with introgression of just a few genes from northern cardinals into Pyrrhexia in the urban environment. The function of these genes, I think, is also very interesting and kind of unexpected. We saw that 2 of those genes, that CH037 included, have a role in vision. And one of them is rhodopsin, which is the light-sensitive protein in rod cells in your eye. So very involved in light vision or night vision, potentially really important for vision at dusk when there might be predators, potentially also involved in circadian rhythms. So selection on their ability to shift their awake schedule. And then also we saw 2 genes involved in muscle function and development that show evidence of introgression, which I think is consistent with our other findings about maneuverability in the city with the feather traits. And then we also found several genes that were positively selected but didn't show evidence of introgression in the 2 species. And these had a diversity of functions from sperm and lung function. Sperm was also further reflected in ctDNA with testicular development. So it could have a role in sexual selection in some kind of way. Cognitive function and cancer and asthma, which is kind of hard to untangle because the way that we know what the functions of these genes is from a lot of research on humans and also from like single cell organism studies in labs that also are like asthma and cancer get a lot more funding in this space. So these are really interesting hypotheses, but at this point, a lot of these are just hypotheses and indications for future research. But the general takeaway is that we see introgression playing a role in these species adaptation to the urban environment, and we identify light as night, energetic challenges, and pollution as potential driving stressors for urban adaptation. And the general takeaways from my whole dissertation that we see widespread gene flow in a clade of tropical species, both within and between species. We see that urbanization differentially affects the distributions of two closely related species with a similar niche, and identify canopy cover and shrub scrub and the way that we're cultivating our urban landscape as potential drivers of that difference. We see that urbanization has complex effects on signaling, heat tolerance, foraging, and maneuverability, and that genes involved in adaptation to light at night, energetic challenges, and pollution have potentially been positively selected, and that introgression is an important evolutionary process. And with that, would love to thank so many different organizations that helped fund this research, that helped complete this research, that helped me meet so many people across Tucson and build the network required to do this research in their yards. And my committee, Dr. Kevin McGraw, Dr. Gru Amdam, Dr. Karen Swayze, and Dr. Scott Taylor, who's here over Zoom. Thank you all so much. And also a massive thank you to everybody in the Tucson community. Here's my mom and me and her friend Kathy and Dave Crockett's backyard. Yeah, really grateful for all of the connections that I made through this process and for all the people who shared with me their stories about why they're interested in helping out with this work and what their relationship with their native ecosystem is too. And yeah, thank you to all of the funders as well. And with that, I will take questions.
Thank you. Well timed, well delivered, really wide reaching, and I think a lot of curious results, and I'm sure things that you all want to ask him questions about. So I'll open up the floor. Online again, feel free to unmute yourself or pass the question through the chat.
How do you quantify color when you do this studies on?
Yeah. So Aaron asked how I quantify color when I'm doing these studies. I took photographs of the birds with a DSLR and then used Photoshop to select the region of interest and looked at saturation, brightness, and hue, which are three metrics that other studies have laid the groundwork for us to use these metrics and see like how each of these different metrics are associated with percent crot or like, yeah, density of carotenoids, etc. Deposition of carotenoids in the feathers. So yeah, photographs and Photoshop and standards. Yeah.
I was just curious, what made you pick the study system for your first chapter? Was it just the amount of data availability, migration patterns, or what made that system a good system for answering your overall question?
Yeah, that's a great question. The question for those on Zoom was what made me choose the study system for my first chapter, the Boobies. In particular, the fact that they hybridize is really interesting, and that there's instances of hybridization in several different instances within the clade. That, yeah, 3 of the species overlap in their regions, which is really interesting to me. Yeah, they're along the Eastern Pacific Ocean, and then all 3 pantropical species overlap too. So there's so much sympatry and hybridization going on that it is like an interesting group to ask that. But I did that work also. I started thinking of that while doing a research tech job with Scott Taylor in Boulder, and his dissertation research was also on blue-footed and Peruvian booby hybridization. So some interesting conversations with him also kind of got me on that track.
Question about the end of your third chapter. The differences in breast quality clarification, are those statistics?
Yeah, so the question was on chapter 3, the differences in plumage. Are they statistically different? So This is Bill Hugh, and we see statistically different, so the A and B are significantly different. So urban is different from urban outskirts and rural, but urban outskirts and rural are not different from each other for Bill Hugh. And then for breast plumage or breast brightness and crest brightness, urban is different from rural, but neither is different from urban outskirts for both species.
Go back to the other one, because there's a super stud in there. It must be the multimillionaire in the city then. Is that significant without that outlier?
I don't know. I don't remember.
So you've got something going on, good.
Yeah.
This is a good question. So, Daniel Hughes asked, do cardinals and pyralexia identify conspecifics visually or through song or a mix of the two? I'm interested because of the noted selection on visual genes.
The question is, do these two species identify conspecifics, or are they choosing to mate with each other based on visual or auditory cues? And I would say it's probably a mix of the two, but we don't have really solid evidence in either direction. Their songs are really, really similar. And yeah, I think that's like a right avenue for future research. Yeah, and he was asking that specifically within the context of selection on the visual genes of the two species in the urban area. That answer well enough, Dan?
Probably.
Kind of going off of that, and I don't know how to phrase, but like, what about the urban environment do you think is selecting against brighter coloration and more growth? It's like you said signaling, mate choice, and such. But like,
Yeah.
how does the urban environment change that?
The question is how the urban environment is selecting against brightness in the plumage. And I'm glad you asked that because it's really complicated. And I've been framing a lot of this in terms of evolution. So selection is like a natural way to infer about this process, but it could also be differences in nutrient availability in the city. And that's what seems to be the case in other cities where birds, where northern cardinals have more carotenoids in their plumage because there's a lot of invasive honeysuckle in urban areas in the Midwest and the East Coast. But we're kind of seeing the opposite trend here. And I think, yeah, again, more research into the like mechanisms underlying this would be really helpful to illuminate why we see this pattern. But yeah, I think also stress seems like a pretty likely reason since we're seeing that both species, again, kind of in contrast to the East Coast where cardinals in the city maybe aren't as stressed. There's a lot of bird feeders and opportunities for them to, yeah, they don't seem to be quite as stressed out in the cities. They are more abundant, where here we are seeing effects on the two species. Could be why we see that trend, but I don't really know why bill and plumage are different because they're developing them at different times in the year. So it could also be related to that. Yeah.
Really great job. I think it's super cool that you work with communities and the people in Tucson kind of like state of so that's super fun. Any key takeaways you learned, like lessons for how to work with communities and do that work?
The question is key takeaways from working with communities and doing the kind of work that requires what I did of building a network of different houses where I can work in their backyards. Ask for help as much as you can would be my first thing because it takes a lot to organize and to communicate with everybody. And I guess don't be afraid to apologize because I also would leave emails unresponded to from people. It's like the middle of field season and you're navigating like 50 different residences. So yeah, hopefully never let anybody feel that I didn't respect their time or their interest. I very much did and was just very busy. So hopefully I communicated that effectively with everybody. And then also, I think potentially like long-term projects that like if you are an early professor in building a project, I think that would, like I now have all of these connections that I think if I were running a lab as a professor would be really helpful for me to hand on to another graduate student. So yeah, leaning into community organizations that already exist too.
a series of Zoom questions. So Adari asked, are there any other traits you wanted to measure, but you were not able to? If yes, which ones am I?
Adari asked traits I wanted to measure, but wasn't able to. I really wanted to look at glucose and ketones in the birds, and I did measure that in the birds that I taught in the field, but because of sample size limitations, we couldn't uncover much of the effects of urbanization on these traits. So ketones and glucose are compounds in your bloodstream that are associated with the diet that you're eating. And because I use museums specimen to expand my data set, they don't have blood in them anymore. It's just the skin of the bird. And would have loved to be able to uncover that. We did see differences between the two species in the ketones and glucose that we collected, but didn't see effects of urbanization likely due to sample size. So that's one. Now that I know that they have differences, or that there seems to be selection on vision, I think I would be really interested in looking at like the retinas and retinal morphologies of the two species and trying to uncover why, but that seems also complicated and hard. And I'm glad I didn't also have to do that, because it's just a decision.
And then Alexis asked, can the difference in beak width also be explained by differences in diet?
Yeah, Alexia asked, differences in beak width, could that be explained by differences in diet? Yes, I am less inclined to think so, because in an urban area, they are feeding a lot of bird feeders, and we see from house finches that beaks are longer and more narrow to help them get at more seeds potentially in a feeder. So I would expect it to be in the opposite direction, and I also, I guess, kind of in reference to Adari's question, would have loved to get samples of even just like their fecal samples and do metabarcoding to see what they're eating and if there are differences in what they're eating. So I think that's a potential thing. And I also haven't observed in the field any major differences of the two in their feeding habits. And there's like one study from 1908 or something, what they found in the stomachs of the two birds. It's not very informative. Yeah.
Apologies if you went over this and I just missed it, but how did you categorize the urban outskirts and rural? Was it population density or like building density?
Yeah.
Because you would think that would help.
So the question was how I characterized urban versus urban outskirt versus rural. And some of that was, so urban is just whether or not they're found in any of the urban areas within this, because these are all binary files. It's either urban or not urban. So that was like the easy stuff of it, but then urban outskirts versus rural was a little more complicated. If you look at this map, a lot of the sites are really close to the urban area, and I know that these birds are probably accessing some of the human development. This is a lot of like suburban outskirts and stuff. And looking at this set of samples and specimens, I was reticent to just call all of these rural. And there was a really big natural break in the data as well, where the difference, like there's a lot of samples clustering close to the city, and then the nearest sample is really far away, and then all of these other samples are also really far away from the urban area. So we classified it based on this really big natural break in the data. I have a histogram, but it's not in this slide. Yeah, which was also like good and bad. I would have loved like a way to make it consistent between this study and other studies, but that was the best way to do it with this data set.
Thank you. Same question. There you go. So Katie asked, what are the most important takeaways from these findings with regards to ongoing conservation efforts, especially in the light of climate change and changing bird ranges?
Katie asked what the most important findings are in the context of climate change and the effects that we see on bird populations. I think maybe one of the big ones is the effect of differences in how we're cultivating our landscapes in the urban area, which is complicated because I also know that big trees also allow for walkable streets for people. But there are definitely trade-offs that should at least be discussed at the community level before we just keep using our water resources for the trees in the area that are not native here. And then also, I think recognizing that the diversity of stressors that are put on these species from human activities are multifaceted, and that it's a process of studying, that like more research is always going to be needed because there are so many distinct species, and I was only looking at 2 of them and uncovered a lot of really specific mechanisms. But without this like species level analysis and just looking again at the community level, we might be missing some of these fine scale details that could be, again, just added into the community discussion in bigger models. Yeah, thanks, Katie.
From, I think Diego, and I don't know who else was in that room, but if you could continue this research, what would be your next most burning question?
Diego and Simona asked if I could continue the research. What would be the next question? Which is a very good question. I think I've seen, and I really wanted to do studies of methylation of the DNA and look at, epigenetics in the city, because we know that, that the epigenome responds more quickly to stressors than variation at the genetic level. And that would be really interesting. It's also, again, complicated because one of the main reasons I chose to go the route that I went is because you can get DNA from blood, but you have to sample the whole bird to get a thorough epigenome study. So I think if I had a long-term network that could give me access to birds that dive naturally, I would love to do the epigenome study, but it's complicated and hard.
I think we're about out of time for public questions. Impressive set, by the way, committee level depth and quality. Frankly, I wish we could end it here, but that's not how it works, unfortunately. But please join me again and fantastic step 1. And yeah, feel free to say your lucks and well wishes. And we'll take about 5 minutes, Danny, and then reconvene here once great. Thanks again for coming and supporting Dan.
We did want to definitely save the chat. We have a lot of